We definitely look at each molecule individually, but with the experience behind us. So as I mentioned, our tenure before, that's a huge advantage. So we remember the molecules that are similar. There are a number of biosimilars where it's not the first time they've come to KBI. So we kind of have an idea of where to go down that path. And that really helps us to accelerate for later clients. The same thing with ADCs. If we know what molecule or method is going to be the trouble method, so I've already said that's the charge heterogeneity, we know to start with that method first in our method development.
Welcome back to part 2 of our conversation with Amanda Hoertz from KBI Biopharma. I'm your host, David Brulman, and in part 1, we explored Amanda's journey and the fundamentals of ADC development. Now we're diving into the nitty-gritty, the analytical puzzles, the formulation challenges, and scaling strategies that make ADCs so complex. Amanda will share KBI's innovative approaches, leadership insights from managing 300 scientists, and the future trends shaping biologics development. Let's jump back in and uncover the secrets of successful ADC commercialization. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brulman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Let's shift our conversation towards the more, I'd say, project management parts or the organizational part. What are some key best practices you follow at KBI to accelerate the path of ADC development?
So the approach is the same whether it's ADC or non-ADC. We found at KBI that's most successful is we have a program manager, and that stays the same for the client. But then we also have dedicated teams in each space. So once you have a dedicated director and group leader, for example, in the analytical space, that will be the same person you work with for the life of the project. It helps because we learn what this customer wants versus that customer, right? This customer may want all the raw data exported and sent to them on a secure share drive so they can make overlays. This other client may not need that. One client may want it all in a PowerPoint presentation. One may not. One may have a critical filing deadline. One may not. So by having a consistent point of contact, and that contact will stay the same if you have another molecule at KBI, which helps because you don't have to relearn that relationship. Everything is consistent. You know how to get what you need. And also you know when they need something. For, let's say, an IND update, you understand who to talk to and you know what they need. It's a good relationship. If the client invests in it and KBI invests in it, it makes everything a lot faster. What we've seen at some CDMOs that we outsource some services to, for example, sterility and CCIT, it's very confusing as to who to talk to. So when we have investigations at those other partner sites and we are responsible for the relationship, I sometimes struggle to get answers out of a director or somebody at a different company. Not going to name names because I don't want to badger anybody in particular, but it's confusing. And I can only imagine as a client, when you have a critical filing for your IND and you need this file now, if you don't know who to contact or can't get a good tree of responsibility, how frustrating that would be. So we try to make sure that it's seamless. I'm not pretending the KBI is perfect. There's definitely times when a client needs something and the timeline is insane and we try to meet it and we do everything we can. And that's one of the strengths of the analytical portion of KBI. So a typical CDMO has a QC team of about 20 to 30 people that execute testing, usually release and some limited stability. KBI has, at the Hamlin site alone, 200 analysts. And then in the network, it's 400, 300 of which are under my supervision. And it means that if you need something as a client, and I can throw basically a flood of people and a flood of instruments on something to make sure you get what you need. It's an army, and we absolutely can destroy any task by rearranging priorities. And that's one of the benefits of having the mammalian network for analytical testing under me and the process development mammalian network under Leslie Wolf at KBI is that we can adjust priorities real time. There's no in-depth conversation or negotiations. And even with our microbial partners, we leverage each other's teams to make sure that we get what the client needs. We have excellent relationships across our entire network.
Having that many people is impressive, and it can definitely be an advantage if you manage your network well or your people well. So my question, Amanda, how do you ensure consistency and the knowledge transfer among this huge army? And what is your framework for building analytical methods that are robust and scalable?
I can't take credit for this because I'm benefiting from my parentage at KBI because they've set this up in a very scalable way. So at the Hamlin site specifically, there's 200 people there. We have 8 directors, and then below the directors are group leaders, below the group leaders are project leaders. So it's a very complicated tree, and that involves the knowledge transfer as well. And basically, we've all been raised. The average tenure at North Carolina AFS is almost 6 years, and the average tenure for managers is almost 10 years. And that is the key to the success. That's atypical for industry. We very much advocate for our people. That's my job, is to make sure that I am doing everything I can for the analysts all the way up to the managers. And those managers do the same. And that inspires a lot of loyalty, a lot of consistency. And that helps because if we're not constantly turning over analysts, we are able to benefit from their knowledge base, and we're able to also do improvement projects. Our analysts are the most turned over, but the average is almost 4 years, which is a little bit different of the industry average, which is around 2 years. So we benefit from that. And I think once you have a good culture, it really is easy to maintain it. When you allow that to degrade is when there's bigger problems. So I can't take credit for it. I'm just trying to maintain what was entrusted to me when I started leading the department. But that's how that many people can all work together. We also all have the same mentality. We know that we need to deliver for the customers. There's no confusion as to our parties, and we have a very clear decision tree. If I talk to one of my directors who's working on a project and I say this other project has reached a critical status and we need to rearrange priorities, first of all, we have a lot of depth to move things around. But second of all, we all understand how that priority is being determined, and it is done. And same thing for me. If my boss, Sigmund Mustafa, who's our chief scientific officer, if she tells me, I need you to make sure this client or this activity is completed, we do it. Very much not a military kind of way, but it's clear. The decision has been made, and we need to now actualize it. So it really helps.
Yeah, that's fantastic. I think clarity is key. And it's great that you can leverage this huge network. So I'm curious, Amanda, well, you mentioned the culture is great and you put a lot of work into the culture. But on the technical side, how has digital transformation changed the way you operate, and how are you leveraging new technologies?
No, that's a great question. Starting with things like a LIMS system and an ELN, we've introduced those so Hamlin is our largest site, though it's always going to be the slowest to be introduced to our new technologies. Right now, currently, we have the LIMS working at our non-GMP sites and then our 2 smaller GMP sites, so our Boulder site, which is microbial, as well as our Geneva mammalian site. And we are implementing the LIMS. As you'd imagine, we have over 650 active stability studies, so bringing those in incrementally from a paper system to an electronic system will take some time. So we're triaging and deciding how we do that. Our target is by the end of 2025 to be fully in the LIMS system. That allows us to instantly understand our statuses, allow us to compile data for clients, allows us to track it in a much more robust way. We have had a very successful paper system, but paper can be damaged. Paper can be lost. Electronics cannot be when they're backed up appropriately. For the ELN system, same story. So it's been implemented at our GMP and non-GMP site in Boulder, Colorado, and we have it at our non-GMP mammalian sites, and we're bringing it on in our GMP mammalian sites now. It'll allow us to link everything a lot faster, and we already give all of our raw data to our clients, so it is literally scanned pieces of papers, but we'll now be able to do that even more efficiently. So they'll be able to get all of the raw data now in electronic format, and we'll be able to link and search for things in a better way than we would if we were dealing with a paper system only. Finally, what we have at our non-GMP site is we've implemented a lot of automation. So TCANs are huge for Derek Ryan, who is our Senior Director of Analytical Development that reports into me in the mammalian network. Their ability to process hundreds and thousands of samples that are needed for PD on a regular basis is because they have those TCANs. We tried implementing a TCAN at our GMP site. The complication is that the audit trail is not straightforward. It's literally code. So what we found instead, and I love Derek's team because they get to test things out in a non-GMP development space, they pulled in the Waters Andrews Plus instruments there. They're not using it with the GMP audit functionality, but it has one, and we purchased those for our GMP site. So the Waters Andrews Pluses are able to basically take away all the repetitive pipetting from analysts. It improves consistency, it's going to reduce our deviations and investigations, and it's going to allow you to walk up, set up the ELISA to be prepared, the dilution series, everything you need, and then come back, take the plate, and just pop it into the plate reader. Important, that has a really clear GMP audit trail that our reviewers are able to audit and understand. So that's a huge advancement. So these options altogether, in addition to, we have, as you'd imagine, a very large amount of formulation development data at KBI, have put that into a database, and we're starting to use that with computer learning to see how we can predict for clients that need a quick and dirty formulation, and they understand it's not optimized, but they only have this amount of budget and this amount of time. How can we set them up with the best chance for success to get that formulation that's ideal? Those things together are all works in progress to be fully implemented, but it's an exciting time to add efficiency and also reduce some of the manual work the analysts have to do.
Before we continue, Biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. Speaking of success, Amanda, what would you say are some of the best practices you follow to accelerate the path of ADC development? And I'd say, what is core to your company culture to enable that?
We definitely look at each molecule individually, but with the experience behind us. So as I mentioned, our tenure before, that's a huge advantage. So we remember the molecules that are similar. There are a number of biosimilars where it's not the first time they've come to KBI. So we kind of have an idea of where to go down that path. And that really helps us to accelerate for later clients. The same thing with ADCs. If we know what molecule or method is going to be the trouble method, so I've already said that's the charge heterogeneity, we know to start with that method first in our method development. We have a number of, in Derek's team, where he starts with the method qualification after the method development, we have a number of platform approaches where we start and then we follow the molecule because it can always be different, but we understand where to start and what levers to pull to be able to improve the resolution here or improve this separation or fully develop the method to understand all the different either charged species or make sure the HMWs are being separated, the peptide map is being separated. We once had a client come to us with a charge method where it was a single peak and they loved it. And we were like, well, you're not separating anything. So we then showed them what they could do with that method. And they didn't love the fact that now they had more than one peak. But for the method to have value, it has to actually separate something.
Yeah, that's a great example.
Yeah, they didn't like that very much, but we got them to where they need to.
I know. It reminds me of some conversations I had in my career. I would say technological advancements are not always welcome. Let's say it like that. I'm not going to name names.
Yeah, exactly. We want to understand, right? Because that's how the differences between batch 1 and batch 2 may be understood, right? If you see a difference in the performance, you want to understand why. And so as the industry in general continues to evolve, we may see different things. We may get better separation. We may now understand what the differences are. And so we have to accept that.
Yeah, definitely. And there's a lot going on and the industry is evolving. So what is your vision of the future of ADCs and biologics in general, the development? What trends do you see?
I mean, it's really exciting. And I love it for patients. So having more targeted therapies and having more options is huge. And so we saw this in the beginning with biologics. In the beginning, IgG1s, IgG2s, they required a much more customizable approach to understand what you were doing. Now it's a platform. As we continue to have more and more ADCs produced, we're going to get more comfortable with how we can produce these more efficiently, more cost effectively, and we can also weed out the winners and losers faster. It's huge. KBI right now has the ability to test non-cytotoxic ADCs at all of our sites, but cytotoxic ADCs analytically at 2 of our sites. What we're investing in is another lab to be able to test GMP, additionally cytotoxic in the United States, and then in our mammalian network to be able to do the process development and analytical development in a safe space. So that's going to require some capital investment from the company, obviously. They're going to be handling large amounts of payload. And then we're still determining whether we would partner with a client for the actual GMP production with a vendor, or if we would actually have those facilities in-house. But we see this as the next frontier. And so we're making those investments to make sure that we grow with the industry. Because I think our experience with the niche molecules coupled with ADCs, we're in the right position to be able to maximize the benefit.
So you have built a successful career leading a huge team now of 300 people. What advice would you give to biotech scientists looking to excel in complex modalities like ADCs? And I would say also in managing quite a big organization that's maybe not at the same site, it's cross-functional and so on.
Well, for managing the site that's this large, it all comes down to having the right people working for you and working with you, because there's no way that I could be responsible for this many people and this many issues that could come up with 300 people. I mean, it has to be, my direct reports have to be able to handle things, and we triage as we go down and as we go up. So that's the most critical part there. When it comes to building a successful career, people don't love change, but I think change is part of what we do. My job has changed over the years. The industry has changed over the years. And so I think absorbing that information and being willing to move with what's hot and what's on the market is important. ADCs can be scary for a lot of people. There's a lot of drugs that we can work on that don't have that cytotoxic payload, that are easier to handle, that are less risk. But this is where the industry is going, so we need to find a way to do that successfully and safely for our patients as well as our employees. So I think that being nimble and being willing to move with the market is critical. When I came to KBI, I only intended to stay for 2 years. And then there was just always, I was in the right place at the right time. I don't think there was a master plan. I think it just ended up being that there were opportunities and I took them and they were offered to me. And I think that a lot of my similar colleagues at KBI have had the same thing. As we've grown, there's been opportunity and it's been a great place to be. But I don't know that you can plan that kind of growth out, and I don't know if it can be replicated. It just comes down to being willing to do what's needed to make a company successful.
Well, this has been great, Amanda. Thank you so much for sharing your passion, your insights. What is the most important takeaway from our conversation?
I think just that ADCs are an exciting new therapy, and there's a significant improvement on our existing therapies that give patients an improved outcome and less side effects. There's such a rich pipeline in industry right now, and being able to adapt to that and characterize these biologics is the next challenge. So we're looking forward to being able to be part of that solution for industry. Thanks for taking the time to talk to me today. I appreciate it.
That's fantastic. Amanda, where can people get a hold of you?
Oh, so there's the KBI website, and there's also the portal, which is the standalone.kbi.bio, which will allow you to get quick quotes, ask questions. It's very interactive and easy. It's like, we call it the Domino's pizza tracker, so you can make a request and see where it is quickly.
Excellent. I'm going to leave all the links in the show note so you have it there. And once again, Amanda, thank you so much for being on the show today. It was a huge pleasure.
Thanks, David. Appreciate it.
What a fascinating conversation with Amanda Hoertz. Her insights on ADC development, analytical strategies, and leadership are invaluable for any biotech scientist. Remember, if you need guidance on process development or any manufacturing challenges you may have, book a free call at brumelconsulting.com/call, and I'd love to help you start. And don't forget to leave us a review on Apple Podcasts or whatever platform you found us on. It will mean the world to me. It truly makes a difference. So thank you so much for that. And thank you so much for tuning in. Until next time, keep doing biotech the smart way. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.